So in this video, we're going to cover mitosis and meiosis. And so just an overview to give you an idea what is covered in this chapter. We'll talk about types of cell division and reproduction. So we'll talk about sexual reproduction versus asexual reproduction. We will talk about the eukaryotic cell cycle and mitosis. We'll talk about meiosis and crossing over, and then finish off with alterations of chromosome number and structure. Mitosis is the cell cycle to produce two exact copies. And so what you start with is a mother cell. And this mother cell is going to duplicate, and it's going to divide into two daughter cells. And so what you end up with is you end up with daughter cells that are identical to the parental cell. So this type of cell division is essential for reproduction, so for single-celled organisms. On the left here for reproduction, we can see an amoeba, which is a single-celled protist that is dividing into two, and it divides by using mitosis. It's just going to make a copy of itself and then divide. That is a type of reproduction that is known as asexual reproduction. Asexual means without a partner. Mitosis is also essential for growth and development. This is used to produce multicellular organisms. So if you think about yourself, you start out as one single cell. Egg gets fertilized by sperm. You end up as a single-celled zygote. That zygote divides into two. Those two cells divide into four, and so on and so forth, until we end up with this trillions of cells organism, this multi-celled organism that the cells in your body arose by mitosis. Now, if you think about it, if mitosis produces two exact copies of the parental cell, if you think about all the cells in your body, would all the cells in your body have the same DNA sequences or different, right? We know we have different cell types. We have skin cells and liver cells and pancreatic cells, neurons, etc. So we know we have different cells, but I want you to think, do those cells have the same DNA sequence? The answer is yes, they do, because they all arose by mitosis, exact copies. What makes cell types different from one another, what makes a skin cell different from a liver cell, from a blood cell, etc., is what we call differential gene expression. It has all the same DNA sequences, it has all the same genes, but what makes two cell types different is what genes are being expressed, what genes are being turned on, and which ones are being turned off. So like in a pancreatic cell, it's going to express or turn on the gene for insulin so that it can produce insulin protein. A red blood cell is going to express the gene for hemoglobin. Those genes are in all the cells in your body, but what makes the cell types different is which genes are turned on or off. And so all the cells in your body, the DNA sequences are exact copies, except if there is a random mutation. This mitosis is also essential for tissue renewal, so replacement and repair of damaged cells. So if you think about if you get a cut, right? If you cut your skin, the skin cells in that area become damaged. But does that cut stay there forever? Answer is no, it's going to heal, because the cells in the area around where the damage occurred is going to undergo mitosis, and it's going to fill in and repair that damaged tissue. So the cell cycle is collectively composed of two big phases. We have interphase and we have the mitotic phase. If we look at interphase, interphase is broken into three stages. We have G1, which is going to be our synthesis of macromolecules and cytoplasmic organelles. So this is where the cell is going to make their proteins, they're going to metabolize their carbohydrates, etc. And also when the cell is going to synthesize their organelles. So in preparation for division, for example, we need to make more of different organelles so that when the cells divide, each cell ends up with a copy of those organelles. S phase. S phase stands for DNA synthesis. This is where DNA gets replicated. DNA gets replicated, it makes an exact copy. And so what you can see is in this picture up here, the one where you can see this one little line, that is one chromosome. And that one chromosome is made of one DNA molecule. When we go through S phase, we are going to duplicate that DNA. We're going to make an exact copy. So notice that this one now has an exact copy. This chromosome is now 2 DNA molecules, and it's held together. These are now referred to as sister chromatids. They're held together at this centromere. So S phase is for DNA synthesis. After the DNA is duplicated, we go through G2. The G stands for gap, so gap 2. This is just a time for the cell to have additional growth. So basically to prepare for division of the cell. Now, in terms of interphase, this is metabolism and growth, and about 90% of the time, the cell is going to be found in interphase. And so this is the period of the cell cycle that lasts the longest. And so if you look at, let's say, an onion root tip under the microscope and you look at that slide, what you'll notice is that most of the cells that you see are in interphase. And that's because interphase takes up the longest amount of time. Then we get the mitotic phase. And the mitotic phase can be broken into 2 parts. We have mitosis. And in mitosis, we are going to divide the nucleus, so meaning we're going to separate the contents of the nucleus, so the DNA. Cytokinesis, cyto refers to cell, cytokinesis divides the cytoplasm. This is where it physically divides from one cell into 2. So in the mitotic phase, we have both mitosis and then cytokinesis. And so again, mitosis is going to separate the DNA. Cytokinesis is actually going to physically separate them into 2 daughter cells. But before we can talk about how mitosis works, we need to discuss how DNA is organized in the nucleus. So the genome is all of the cell's genetic information, and a eukaryotic genome is broken up into chromosomes. Each chromosome is one long DNA molecule. Now, you might recall when we compared prokaryotic and eukaryotic cells, prokaryotic chromosomes are circular. Eukaryotic chromosomes are linear. They're long pieces of DNA. In our cells, our chromosomes, we have 46 chromosomes. So we have 46 long linear DNA. So what we're looking at in this particular cell, this is a nucleus of a cell in yellow. We have a black, a blue, a red, and a green. Each one of those represents 4 chromosomes. And each of these chromosomes is a long DNA molecule. Now, I didn't just draw them as straight lines because in actuality, they're going to be compacted and folded and organized within the nucleus. So each of those different colors represents its own chromosome. So this chromosome has a sister chromatid in a minute, not yet. But what you'll see is that this DNA is one long piece of DNA. So nucleus has 4 chromosomes and 4 long DNA molecules. Now in S phase, remember that in S phase, S stands for synthesis, and that means that that is when the DNA is going to be duplicated. It's going to copy itself. So if we look at our example of our nucleus, notice that our black one was duplicated, the blue one was duplicated, the red was duplicated, and the green was duplicated. So now this nucleus has 4 duplicated chromosomes. At this point, when they're held together at what we call the centromere, which we'll see in a minute, when those DNA molecules are held together, they are considered one chromosome. So each of those is one chromosome. So the black one collectively is one chromosome. But that one chromosome is 2 DNA molecules. So it is one chromosome, but that one chromosome is made of 2 DNA molecules. So if we look at this nucleus, it has 4 chromosomes, but it has 8 DNA molecules. As long as they're held together at the centromere, it is considered one chromosome. So duplicated chromosomes are attached at the centromere. That's the attachment point between those duplicated chromosomes. And the centromere consists of special proteins. And these proteins recognize and bind to specific DNA sequences. And when they bind to those sequences, they're going to hold that DNA together. What we get is that they're referred to then as sister chromatids. Sister chromatids are each DNA molecule of a duplicated chromosome. And so this green one and this green one, those are both sister chromatids. And again, they're held together at this centromere. It is only considered to be one chromosome as long as it's held together at the centromere. As long as it's held together at the centromere, it is called one chromosome. So chromosomes need to condense and pack in when the cell goes to divide. So if we look at a cell that is in interphase, the DNA is not tightly packed in. It's loosely associated. And when it's loosely associated, it's referred to as chromatin. Under the microscope, you don't see that characteristic chromosome X-looking structure that you might think of. Instead, it just looks like a tangled mess. The chromatin is basically going to be a collection of DNA and proteins. And remember that eukaryotic cells, the DNA is wrapped around histone proteins. And so this helps to package in the DNA. Now, in interphase, the chromosomes are decondensed. They're not packed in very tightly. However, when the cell gets ready to divide, when it goes through mitosis, one of the things that's going to happen is that during prophase, the DNA needs to condense and pack in more tightly. And so you can think of it like if you had a big ball of yarn, right? If I have a big ball of yarn and I try and separate it, when it's in a big ball, it's going to be difficult. Things are going to get tangled, right? It's going to be a big mess. So the easiest way to separate it would be to organize it. Think about for thread, you would wrap it around a spool, and that allows it to be organized so it's easier to separate. Same thing with the chromosomes. The chromosomes need to condense and they need to pack into that typical X-looking chromosome structure that we're used to seeing, because that then allows it to be separated easier before when the cell divides. So if we look at what happens to chromosomes during mitosis, so again, in G1, each chromosome is one DNA molecule. So G1, remember, comes before S phase. S phase is where we get DNA synthesis. Chromosomes are going to duplicate. So in S phase, we end up with our sister chromatids. And so now when they duplicate, these are going to be sister chromatids. Those sister chromatids are exact copies. They are held together at the centromere. The centromere, that's what's going to hold those two DNA molecules together. Again, as long as they're attached at the centromere, that would be considered one chromosome, but two DNA molecules. Now, as the cell goes to divide during mitosis, that is when the chromosomes are going to pack in very tightly. They're going to condense, and that's going to help prevent against tangling. When we go through mitosis, remember that the goal of mitosis is to make two daughter cells that are identical to the parent cell. The way that that is accomplished is that during mitosis, those sister chromatids, those exact copies of one another, are going to separate. And so when they divide, you're going to end up with two cells. Each of those cells has one DNA molecule. It has one chromosome. Notice that when it's finished, it looks the same as when it started. So we went from one cell to two cells, and they're exact copies of the parental cell. So for the questions for this lecture, I went ahead in the video and I numbered them questions 1 through 9. And what I want you to do is there will be a participation assignment on Canvas that you will need to write number 1 and then whatever color you think the correct answer is. And after the due date has passed, I will post the answers for you to study to make sure that you get the right answer. So let's go through and talk about what are these questions. So first question, there are blank chromosomes in the cell and blank sister chromatids. So I want you to take a minute, pause your video, and when you're ready, push play to see the answer. So let's look at this. So if we look at this, how many chromosomes do we have? We have 6. We have 1, 2, 3, 4, 5, 6. We have 6 chromosomes because they're held together at centromeres. And blank sister chromatids or blank DNA molecules. Answer, that you have to post. So I helped you with the first part, but you need to go on and post your answer for this one. So the mitotic spindle consists of 2 centrosomes and microtubules. So they are these little T-shaped structures, and these centrosomes are in animal cells made of centrioles. They are these structures that are made of microtubules. But centrioles are not necessary. Plant cells don't have them, yet they still can form a mitotic spindle. The goal of the spindle and the goal of the centrosome, so the centrosome's job is to act as what we call the microtubule organizing center. Its job is to organize those microtubules. So assembly of the spindle is going to start at the centrosomes and it's going to radiate out. The single centrosome is going to replicate during interphase so that we have 2. They will end up so that there are 1 at each end. So now we need to get back to the mitotic phase of the cell cycle. We talked about the structure of DNA. Now we need to talk about how it's actually divided. So let's walk through the steps of mitosis. The first phase of the cell cycle is going to be interphase. And in interphase, remember that the chromosomes are not packed in tightly. They are at this point referred to as chromatin. So the chromatin, again, is going to be the DNA wrapped around those histone proteins. We also, within the nucleus, have a nucleolus. Remember that the nucleolus is the site of ribosomal RNA synthesis, as well as the assembly of the ribosome. So we have our DNA, it's contained within our nuclear envelope. Now, in the cytoplasm during interphase, the cytoplasmic contents double. So the organelles are going to double, etc. 2 centrosomes are going to form. So notice that we have our 2 centrosomes. In the nucleus, chromosomes are going to duplicate during S phase. Nuclei, the site of ribosomal assembly, is visible at this point in the cell cycle. And so what you're looking at the top is that we're looking at different structures within the cell. So the blue is staining the nucleus, and that green, or what looks kind of yellowish, those are the microtubules that are fluorescently labeled. So what we're going to do as we go through this lecture is I posted a mitosis study sheet, and you want to fill in the number of chromosomes and the number of DNA molecules at each step of this process. So what I did is I went ahead and I drew out the stages of mitosis, and I simplified it so that I only drew 1 chromosome. So notice that when we look at G1, I have 1 chromosome from mom. I color coded that pink, and 1 chromosome from dad. Because of our 46 chromosomes, they are arranged into 23 pairs. Now, I didn't want to draw 23 pairs because it gets very busy and difficult to see. So I simplified. I drew 1 from mom and 1 from dad. So in this cell in G1, it has 2 DNA molecules, right? So 2 DNA molecules and 2 chromosomes. So if we're thinking about in humans, in G1, how many DNA molecules do we have? 46. How many chromosomes do we have? 46. Because at this point, every chromosome is 1 DNA molecule. Now, the cell is going to go through S phase, and then G2. It will look the same at both. So remember that what's going to happen in S phase is that chromosomes are going to duplicate, and we're going to end up with sister chromatids. So the pink one is going to make an exact copy. The blue one is going to make an exact copy. So at this point, if you look at this cell, the drawing, there are 4 DNA, but still 2 chromosomes. There's still 2 chromosomes because as long as they're held together at the centromere, it's still considered each one is still 1 chromosome. But each chromosome is 2 DNA molecules. So now in S phase, if we have duplicated our DNA, how many DNA molecules do we have? 92. So 46 and 46 is going to be 92. How many chromosomes do we have? Still 46. It's just that at this point, each chromosome is made of a duplicated DNA. So it has sister chromatids, and so each chromosome is now made of 2 DNA molecules. Next, we come to prophase. And in prophase, in the cytoplasm, the microtubules begin to emerge from the centrosome, and the spindle begins to form. So those microtubules are going to form. The centrosome begin to move to opposite poles of the cell, meaning they're going to start to move to opposite ends. In the nucleus, their chromosomes coil and become compact. So this is where they're going to start to wrap very tightly around those histone proteins, and they're going to fold back on themselves. And so if you look in the nucleus of this fluorescent cell up here, you can start to see these little squiggly lines in the nucleus. Those are going to be the chromosomes that are beginning to compact. During prophase, the nucleoli also disappears. You can see that the mitotic spindle is forming, and the centrosomes are beginning to move towards opposite poles. The next phase is going to be prometaphase, which is also known as late prophase. So for the purpose of the table that we're going to fill in, we're going to link these with the prophase. So in late prophase or prometaphase, spindle microtubules reach the chromosomes where they will attach at structures called the kinetochores. And the kinetochores are on the centromeres of the sister chromatids. And the spindle fibers will be responsible for moving chromosomes to the center of the cells through associated protein motors. And so these motor proteins will walk along the microtubules and carry the chromosomes with them. Other microtubules meet those from an opposite pole. We call those the non-kinetochore microtubules. So if you look at some of these other microtubules, these are our non-kinetochore microtubules. Their job is not to move the chromosomes, but instead to help elongate the cell. During prometaphase, the nuclear envelope is going to break down. You can see in this fluorescence image, you can see your spindle. So notice that your centrosomes are at opposite ends of the cell. You can see spindle fibers. And the blue, you can now start to see the chromosomes really starting to condense. They're no longer in a nucleus as well. So if we go back and we continue on in our table, so notice that if we look at prophase, we drew the nuclear envelope starting to break down. You can see the nucleus has now become a dotted line. We can see that the centrosomes, the brown, are starting to move to opposite ends. We have our spindle fibers forming, and we still have our sister chromatids linked together. Again, the pink ones are from mom, the blue ones are from dad. In this example, there are 2 chromosomes, right? The 2 pink together are 1 chromosome. The 2 blue together are 1 chromosome. So we have 2 chromosomes, but those 2 chromosomes are made of 4 DNA molecules. So in prophase, in humans, we still have 92 DNA, and we still have 46 chromosomes, right? Our chromosome number doesn't change, and they're still duplicated, which is why the DNA number is 92. Then we come to metaphase. In metaphase, the spindle is fully formed, and the chromosomes are going to line at the cell equator in the middle. Think of metaphase middle. They align on this imaginary plate that's known as the metaphase plate. The kinetochores of sister chromatids are facing the opposite poles of the cell. So one sister chromatid is facing one side, facing one centrosome, and the other sister chromatid is facing the other centrosome. And so you'll notice when you look in these drawings, it kind of represents it like they're lined up perfectly in the middle. But if you actually look under the microscope, you'll notice that the chromosomes are not perfectly straight up and down in a line. And that's because the kinetochore is anchored in the middle, but the arms of the chromosomes can go off to the side. It's like if you think of your body, right? Your torso is one piece, but then you have your left arm and your left leg and your right arm and your right leg, and those could go off to the side. They're not anchored in the middle. They can go either way. So when you look for a cell that's in metaphase, don't necessarily look for it to look perfect. What you're looking for is that the chromosomes are lined up at the metaphase plate. So if we look at our drawing for metaphase, notice that our chromosomes are lined up at the metaphase plate. We have one centrosome, two centrosomes, they're at opposite ends. And then when we look at our chromosomes, the sister chromatids face opposite ends of the cell. One sister chromatid faces one side, one faces the other. We still have 2 chromosomes and 4 DNA. All we've done at this point is move them in the cell. So in humans, we still have 92 DNA and we still have 46 chromosomes. In anaphase, sister chromatids are going to separate at the centromere. So now those sister chromatids are going to separate. They're going to go to opposite poles of the cell. Once those sister chromatids separate and they're no longer held together at the centromere, they are now separate chromosomes. Whereas before, each chromosome was made of 2 DNA molecules. Now, those 2 DNA molecules have separated and temporarily they're 2 separate chromosomes. As the chromosomes move in the cell, they're going to move because there are motor proteins that move the chromosomes as they walk along the mitotic spindle. And during this part of the cell cycle, the kinetochore microtubules are going to shorten. And when they shorten, that is going to pull the chromosomes to the centrosome. And so remember when we watched the video that the spindle microtubules or cytoskeletal elements are very dynamic. They move, right? They grow, they shorten, they grow, they shorten. And in this case, they'll shorten to help pull the chromosomes to the ends of the cell. The cell will elongate due to the lengthening of non-kinetochore microtubules, the microtubules that are not connected to the chromosomes. So all of these yellows that are just attached to each other. So if we go back and we look at our drawing, so here we have our cell in anaphase. Our sister chromatids have separated. They've gone to opposite ends of the cell. Our DNA number is still 4, right? If we look at metaphase, the number was 4. It's still 4. We still have the same number of DNA. However, when sister chromatids separate and they're no longer held together at the centromere, notice that temporarily our chromosome number goes up to 4. So if we fill in our table, number of DNA molecules in anaphase is still 92. We haven't changed our DNA number, but we have changed our chromosome number. Every DNA molecule is now its own chromosome, so now we have 92 chromosomes. Then we come to telophase. And in telophase, the cell continues to elongate. The nuclear envelope is going to start to reform around the chromosomes at each pole, establishing the daughter nuclei. The chromatin is going to uncoil. It's going to start to unpack again. Nucleoli is going to reappear and the spindle disappears. So for me, the way that I remember what happens in telophase is the exact opposite of prophase. So the stages in order, I just think of PMAT, prophase, metaphase, anaphase, telophase. In prophase, chromosomes condense. They pack in tightly. Telophase, they decondense. They unpack. Prophase, nuclear envelope breaks down. Telophase, nuclear envelope reforms. Prophase, nucleoli disappear. Telophase, nucleoli reappear. Prophase forms spindle. Telophase, spindle breaks down. So literally they are the exact opposites. And so if we look at the fluorescence image of the cells undergoing telophase, you can see that the chromosomes have migrated to opposite poles of the cell. You can see that they're starting to decondense, right? They're not packed in very tightly. You can also see that they're starting to go back in kind of a circular shape because the nucleus is going to reform. And you can kind of see this little white line, this place in the middle of the spindle where you don't see the spindle. That's because one of the things you're going to see is that while the cell's undergoing telophase, simultaneously it's also undergoing cytokinesis, the division of the cytoplasm. And so let's look at how that happens. Cytokinesis is how we divide the cytoplasm into separate cells. In animal cells, the way that cytokinesis works is through what's called a cleavage furrow. And a cleavage furrow is going to form by a contractile ring of microfilaments, or actin. Remember that actin works with their motor protein, which is myosin. Actin and myosin contract. Think of that they're used for in muscle contraction. And so actin and myosin, when they contract, they're going to squeeze and they're going to pinch that cell into two. So this ring is going to form around the outside. Imagine you had like a water balloon and you were able to tie it off in the middle. That's kind of like what this cleavage furrow is going to do. It's this ring of actin and myosin around the cell, and it's going to pinch and it's going to divide the cell into two. So if we go back to our drawing, we have our cell divided into two, right? So telophase and cytokinesis, we're putting them together. And so what you can see is that when the sister chromatids separate, they went to opposite cells. So we end up with two daughter cells. And notice that those daughter cells are exact copies of the parental cell, meaning the cell in G1. At this point in telophase, we are back to 2 DNA and we're back to 2 chromosomes. So in the case of our human numbers, right, in our human numbers, if we were at 92 and 92 and sister chromatids end up in opposite cells, we have 46 DNA now and 46 chromosomes. And again, you can see that the numbers are going to be the same as they were in G1. Because again, the goal of mitosis, make an exact copy. That is how you can tell if you filled in this table correctly. You should end up with the same number that you started with. So if we look at cytokinesis, cytokinesis again divides the cytoplasm. This is just comparing and contrasting animal cells and plant cells in the way that they do cytokinesis. They do cytokinesis a little bit different because plant cells have a cell wall, animal cells do not. So we talked about animal cells using a cleavage furrow. So again, we have our microfilaments or our actin forming a ring around it with its motor protein myosin. And it's going to contract and it's going to pinch and it's going to divide the cell into two. Plant cells, on the other hand, can't do this because plant cells have that rigid cell wall. So we can't just use actin to squeeze because that plant cell wall is very rigid. So the way that plants do cytokinesis is a little bit different. They're going to form a cell plate. And so what's going to happen is we're going to get these vesicles. Remember we talked about like transport vesicles, for example. So these vesicles are going to go to the middle of the cell between the two nuclei that have formed. And eventually those vesicles will start to fuse and we will get a structure known as the cell plate. The cell plate will continue to elongate, and when it elongates, it will attach to the cell wall, and eventually we will end up with a new cell wall. So, question for you. This is question number 2 for you to fill in on Canvas to get credit for the participation. If a cell is not dividing and has not yet replicated its DNA, duplicated its chromosomes, what phase of the cell cycle would it be in? Red, gap 2, yellow, synthesis or S phase, green, G1, blue, prophase, or purple, the mitotic phase. So if you need to, pause your video, write down your answers so that you can post it on Canvas, and after the due date closes, I will provide you with the answers. So next question, question number 3. What phase of mitosis is this cell in? Is it red, prophase, yellow, metaphase, green, anaphase, blue, telophase? And again, you will post this answer on Canvas. The next thing that we're going to do is I'm going to show you a video summarizing mitosis.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
So you're seeing the chromosomes condense, the nuclear envelope starting to break down. The chromosomes are going to begin to line up at the metaphase plate. And in a minute, you're going to see sister chromatids begin to separate. So they're lining up in the middle still. There they go. Sister chromatids separate. We're starting to see cytokinesis starting to pinch off, and the chromosomes are going to go back into the nucleus and decondense. And so that's a time-lapse photography showing mitosis in action. So now we're going to talk about control of the cell cycle. So not all cells divide all the time. The number of times a cell divides depends on the cell type, the conditions within the cell, the intracellular conditions, as well as the conditions outside the cell, the extracellular conditions. And so there's a lot of signaling involved that tells the cells whether or not they should divide. Human skin cells, for example, divide very frequently, right? We lose our outer layers of skin, our lower layers undergo mitosis, and they repair and build the skin from the inside out. And so human skin cells divide frequently. Liver cells can divide, but only do so when the liver is wounded. And nerve and muscles in the mature human never divide. So now we're going to talk about what controls the decision to divide or not. How does the cell know whether or not it should divide? Well, in the cell cycle, there are what we call checkpoints. And these checkpoints you can think of as stop and go signals. Basically, they're like, think of a traffic light. If a certain condition is favorable, it's going to get the green light and it's going to go to the next phase. If conditions are not favorable, it's going to get a red light and it's going to stop and it's not going to continue to divide. And so there are at least 3 checkpoints in the cell cycle. So we have what's called the G1 checkpoint, and the G1 checkpoint allows entry into S phase or causes the cells to leave the cycle and enter a non-dividing G0 phase. So basically this is going to control if the cell is going to be able to proliferate, which means to divide, or if it's going to go into quiescence, which means a non-dividing stage. We also have a G2 checkpoint. And notice that before G2, we have S. So why is the cell have a checkpoint here? Well, the cell is going to make sure that the DNA is duplicated before it proceeds to mitosis, because if the cell hasn't duplicated its DNA and it doesn't have sister chromatids, it can't divide into 2 cells. We also have an M checkpoint. M checkpoint is to make sure that chromosomes have lined up properly and are beginning to separate properly. And so these stop and go signals allow passage through the checkpoints, and they're basically regulated by both internal and external signals via signal transduction pathways. So certain growth factors, for example, can stimulate the cell to divide, or other factors might tell the cell not to divide. And that can either be internally or from external sources as well. So there are several factors that control cell division. One, presence of essential nutrients, right? The cell needs those nutrients if it's going to be able to divide, because it needs it to be able to make ATP for energy. Growth factors. Growth factors are basically proteins that stimulate division. So if growth factors are present, the cell can divide. Presence of other cells causes what's called density-dependent inhibition, meaning that when cells start to pack in tightly, they will receive a signal that tells them to not divide. And that basically you're going to see in a minute is to protect against cancer. Cancer cells lose that density-dependent inhibition. They start to just pile up. They don't get that signal to stop, and they just pile up and they end up forming tumors. And then lastly, we have contact with a solid surface. Most cells show anchorage dependence, meaning that they have to be attached to some surface in order to divide. So this is just showing you about growth factors, right? So if we took some cells, some fibroblasts, and we were to put them on a plate, and one plate was given platelet-derived growth factor, PDGF, and the other was not given PDGF, what you'll see is that the one without the platelet-derived growth factor will fail to divide, while the plate that's given the platelet-derived growth factor will proliferate and will divide, because that growth factor is going to be there to stimulate growth. It tells those cells to divide. In terms of density-dependent inhibition, so cells will anchor to a solid surface, and when the cells have formed a single layer, they will stop dividing. That is their density-dependent inhibition. However, if you scrape some of those cells away, and now they are not in contact on every side, the cells in the area will recognize that those cells are removed, and they will divide and they will fill in. And they will fill in until they have a single layer. That is referred to as density-dependent inhibition. The density is going to inhibit the cell from dividing. Now again, in cancer is uncontrolled cell division. And in cancer cells lose their density-dependent inhibition. They don't care that other cells are around them. They bypass those checkpoints. They're told to divide all the time, no matter what the condition. And so cancer cells will lose this density-dependent inhibition. In terms of anchorage dependence, different cell types have different levels of anchorage dependence. Some just need a solid surface to grow on. Others might even require a certain cell type in order to grow on. And so what you're looking at here, this is actually a picture from my research. And what you can see, those long, thin, spindly cells, those are a type of cell called a fibroblast. And what we're culturing is these are mouse embryonic stem cells. And so we isolated embryonic stem cells from mice, and then we grew them on a dish. Now, embryonic stem cells not only grow on surfaces by themselves, but they also require that feeder layer. They need those fibroblasts in order to attach and in order to divide. And so this is an extreme example of anchorage dependence. It not only needs a surface, but it also needs a certain cell type there to help with their division. And so these little circles that you're seeing here, those are what we call colonies of embryonic stem cells. So the embryonic stem cells tend to grow in these clusters, and those are the embryonic stem cell colonies. Now, cancer cells escape controls in the cell cycle. Cancer cells divide rapidly, often in the absence of any growth factors. They spread to other tissues through the circulatory system. Their growth is not inhibited by other cells, and tumors can form. If we call a tumor benign, a benign tumor will stay at its original site, whereas a malignant tumor will spread to other locations through a process referred to as metastasis. And so if you get a cancer diagnosis, for example, they will have it in stages, stage 1, 2, 3, and 4. 4 being bad, meaning it's metastasized to multiple tissues in the body. 1 meaning good, it's localized to just that region. And so different types of cancers have different prognosis as well as what stage they're at. Best case scenario, you want to have cancer cells that are localized to a particular region, but that have not invaded the surrounding tissue and have not spread using the circulatory or the lymphatic system. So how does cancer arise? 2 things have to happen in order for cancer to arise. And that is that their accelerator must get stuck and their brakes must fail. So think of it like a car. So the accelerator gets stuck, the go signal is stuck, and the brakes don't fail. What normally tells the cell not to divide no longer works. So the stuck accelerator genes are what we call oncogenes. Think of a cancer doctor. That would be an oncologist. So oncogenes are basically genes that normally induce cell division. So basically these are genes that normally tell the cell to divide. So when they're not cancer causing, they're called proto-oncogenes, but when they mutate and they become cancer causing, we call those oncogenes. And so the accelerator gets stuck and the cell to divide is always there. And so that causes cell growth to become hyperactive. The cell is going to divide all the time. Now, that alone is not enough to cause cancer because we have checks and balances. We also have a group of genes which produce proteins that tell the cell not to divide. And those brakes that tell the cell not to divide, those are referred to as tumor suppressor genes. So just like the name suggests, right? Tumor suppressor, they're there to suppress tumors because these genes produce proteins that tell the cell not to divide. And so in order for cancer to arise, you need to get mutations in both sets of genes. You need to get the oncogenes that get mutated to tell the cell to divide all the time, and you need the brakes to fail. You need to get a mutation in the tumor suppressor genes so that they don't have a way not to divide. So those tumor suppressor genes would normally say, stop dividing, but if they're mutated, the cell no longer gets that signal. And if the oncogenes have become activated, now they're being told to divide all the time, and there's nothing telling them not to divide. And so that's going to lead to cancer. Now, usually it takes many genes to become mutated in order for cancer to occur. This is why cancer is often a disease of the middle-aged and elderly. It's more common in older individuals because you need to acquire those mutations in order for cancer to arise. So if you know anything about breast cancer, if let's say your mother had breast cancer and your aunt had breast cancer and other females in your family had breast cancer, that would likely indicate that there could be a genetic component. And there are genes that are involved in this. They're called BRCA1 and BRCA2. And these genes, if they're mutated, it doesn't mean 100% of the time you are going to get cancer. However, having these mutations will increase your risk. Because again, if you think about it, if you need to get multiple mutations to get cancer, if that's already mutated, you're already one step down that pathway. You have less number of mutations to acquire to get cancer to occur. And so that's why, for example, some people who have mutations in the BRCA genes, they might have a preventative mastectomy. They might remove breast tissue to try and prevent against getting breast cancer, because if you don't have that breast tissue, you can't get breast cancer. And so some people will take a preventative approach if they have the BRCA gene. Again, it doesn't mean that they will 100% get breast cancer, but the chance goes up dramatically if they already have that mutation. In terms of cancer treatments, so localized tumors that are in a particular region can be treated with surgery or radiation, right? Because we can either remove the tumor depending on where it is or use radiation that targets that particular area. For tumors that are metastatic, meaning they've metastasized or moved to other locations, generally this would be drugs that prevent cell growth through a variety of mechanisms. For example, they could be drugs that target microtubules, because remember that the mitotic spindle is responsible for moving chromosomes in the cell. They could be drugs that target DNA synthesis, because if you inhibit DNA from replicating, the cell can't divide. Remember that G2 checkpoint? So without the cell duplicating its DNA, the cell can't divide. And then there are a whole host of other types of treatments that are now available for cancer as well. So this is just showing you an example of how cancer progresses. So again, we get a single cancer cell that has acquired enough mutations to lose those checks and balances that tell them not to grow. And that one cell that was growing out of control is going to divide, and it gives rise to a tumor. Now, if that tumor starts to invade neighboring tissues, and then it gets into the blood or the lymphatic system, now those cancer cells can circulate through the blood and might start to go to other tissues, and the cancer cells will start to develop in other tissues as well. And so you could have somebody with lung cancer, for example, where the original cancer originated in the lungs, but it might end up in the brain and other parts of the body as well. So question number 4 for you to post on Canvas. The cell cycle is linked to cancer because cancer always entails red, too much time spent in S phase; yellow, a slowdown in mitosis; green, unrestrained cell division; or blue, a misalignment of chromosomes. So put your answer in Canvas, and then I will post the answers later. Now we're going to move on and talk about meiosis. So to compare and contrast mitosis and meiosis, mitosis's job is to make exact copies. We want to go from one daughter cell to 2 exact copies. Meiosis serves a different purpose. Meiosis is used for sexual reproduction, meaning reproduction that involves 2 partners. So let's say a woman has an egg cell, and if you think about the cells in your body, you have 46 chromosomes. So if a woman's egg, which are her gametes or her reproductive cells, if that egg still had 46 chromosomes, and the sperm, which is the male reproductive cells, the gametes, if the egg had 46 and the sperm had 46 and those came together, you end up with offspring with 92 chromosomes. That doesn't work. So meiosis is used for a different purpose. It's used to reduce chromosome number in half. And so this will result in the halving of chromosome number. This is used for meiosis for the reproduction that involves the fusion of gametes. Basically, the reproduction that involves 2 parents. This is sexual reproduction. And so we have to reduce chromosome number in half so that when those 2 gametes come together, you restore the diploid number. You restore the normal number. Mitosis, on the other hand, no halving of chromosome number, because again, the goal is to make two identical daughter cells to the parent. This is for reproduction without the fusion of gametes. So mitosis is used for asexual reproduction. This reproduction involves one parent. Again, it's simply going to divide and give rise to two daughter cells. Mitosis will result in offspring that are genetically identical to their parent, because again, they arose by simply making a copy and dividing. Meiosis results in offspring that are genetically different from each other and from their parents. So if you think about you and your siblings, unless you're an identical twin, you are not identical because of meiosis and because of the genetic rearrangement that occurs during meiosis. And we'll learn more about that diversity in this lecture. So again, this is just showing you that you're going to have this diversity in terms of sexual reproduction. You will have offspring that look different from their parents. You will have siblings looking different from one another because of meiosis. So in order to understand meiosis, we need a little bit of information about chromosomes. So first, let's start with somatic cells. Somatic cells are all the cells in your body that are not used for reproduction. So a skin cell, a liver cell, a neuron, those are all somatic cells. And so in somatic cells, so all the cells in your body not for reproduction, you have pairs of chromosomes. So 46 chromosomes total, and your 46 chromosomes are arranged into 23 pairs. Those pairs of chromosomes are referred to as homologous chromosomes. And so when you look at a homologous pair, one comes from mom, one comes from dad. So in this case, again, the pink is the one that comes from mom, the blue is the one that comes from dad. Now, how do we know which ones are homologous chromosomes? They're matched in length, meaning they're the same size. You'll see when I show you a karyotype that the different pairs of chromosomes have different sizes, but a homologous pair is going to be matched in length, matched in banding pattern when you look at a karyotype. This has to do with where the chromosome is more condensed or loosely condensed. You get this banding pattern. The centromere position, remember that the centromere is what is going to hold sister chromatids together. The centromere does not have to be at the middle of the chromosome. Centromeres could be off to one end. But on a homologous pair, the centromere is in the same location. Homologous chromosomes also have the same gene locations. So let's say we were talking about the gene for, let's say, eye color, for example, and we'll look at the letter B. So at this position, that is where we have gene B, the gene for eye color. Now, notice that on the one from mom, it has big B. On the one from dad, it has little b. So maybe mom gives the brown eye gene, and brown eye is dominant over blue eye. We'll learn about genetics in the next chapter. So the dominant gene is given the capital letter. So notice that on the sister chromatids, right? Remember that sister chromatids are exact copies from one another. So at that position on the sister chromatids, you're going to find the same letter. It's big B and big B. If we look on the one from dad, the homologous pair, notice that the same position, the same what we call locus, which is the same position on the gene, on the chromosome, at that same position, we have the gene from eye color, but that gene could be a little bit different. It could code for making blue eyes, for example. The alternate versions of a gene we refer to as an allele. So the allele for eye color is at the same position on both of the homologous pairs, the one from mom and the one from dad. And so when you look at homologous pairs, the locations of the genes are going to be the same. So notice that A's are in the same position, B's are in the same position, D's are in the same position, so on and so forth. And so this is just to give you an idea of what are homologous chromosomes. So pairs of autosomes have the same size and genetic composition. The autosomes are the chromosomes that are not sex determining, meaning they're everything excluding X and Y. Human sex chromosomes, X and Y, differ in their size and genetic composition. You'll see this in a minute that the X chromosome is much larger than the Y chromosome, and they have different genes, which is how we have different genders. So a little review. Humans have 46 chromosomes. How many homologous pairs does that represent? So how many homologous pairs does that represent? Answer, 23. How many pairs are autosomes? 22, because the remaining pair are going to be sex chromosomes. So we have one pair of sex chromosomes. So of the 23 pairs, 22 of those pairs are the autosomes, the ones that do not are not used for sex determining, and you have one pair of sex chromosomes. So this is just showing you a human karyotype. And so this can be done by taking a blood sample and fixing the cells so that they're in mitosis. Because remember that the chromosomes are condensed in mitosis, so we can use drugs to fix them at M phase, metaphase. And that is when the chromosomes will be compact into their typical chromosome structure that we think of. So notice that this karyotype is arranged into pairs. Chromosome 1, for example, is this large one, then 2. Notice all the way down to chromosome 22, we have 22 pairs of autosomes. So you'll notice that these chromosomes have different sizes. They also have different centromere positions. So if you look at where the centromeres are, sometimes like for chromosome number 3, it's kind of towards the middle. For chromosome 12 or 18, it's kind of off to one side. And so remember that the homologous pairs are matched in size, banding patterns. So look at the regions where you have darker and lighter parts, the position of their centromere, etc. Now we would know that this karyotype is female because it's going to have 46 chromosomes, 23 pairs, including XX, right? Because in the case of females, the sex chromosomes are going to be XX. So we know that this is a female because it has 2 X chromosomes. Males, on the other hand, males are going to have 46 chromosomes. Now, of their 46 chromosomes, they have 22 pairs that are autosomes, meaning they're not used for sex determination. And then if we look at their sex chromosomes, they have an X and a Y. Notice that the X is much larger than the Y. So their pair of sex chromosomes are not identical in size, yet during meiosis, you'll see that X and Y will behave as though they are a homologous pair. So let's take a look at what we call ploidy. If we call a cell haploid, haploid, think half. It has half its chromosome number. This is a cell with only one set of chromosomes, meaning it does not have its homologous pair. So one of the easiest ways to determine if a cell is haploid or diploid is look for, do they have their homologous pair? If they do not have their homologous pair, they are haploid. What cells would be haploid? Well, gametes. Egg and sperm are haploid. They have half their chromosome number because during fertilization, when you put those two back together, it's going to restore the number of chromosomes. So if we talk about a human gamete, for example, N equals 23. That gamete would have 23 chromosomes because it doesn't have both the ones from mom and dad. Diploid, on the other hand, diploid would be di means 2. This is a cell with 2 sets of chromosomes, meaning it has its homologous pair. You look for the homologous pair. If you see the homologous pair, it is diploid. Which cells in the body are diploid? Human somatic cells. Remember all the other cells in your body. All the other cells other than the gametes are going to be diploid. So what we represent this as is human somatic cells. We say 2N because N is equal to the number of haploid chromosomes. So 2N equals 46. So that diploid cell will have 46 chromosomes. Now, if we look at these examples on the bottom that have on the left, notice that we have homologous pairs, right? They're matched in size, centromere position, etc. So notice that I circled the homologous pair so that you could see which ones were paired. So the two with the red circle, those are a homologous pair. Same size, same position of the centromere. So the pink one represents the one from mom. The blue one represents the one from dad. And then we have this blue chromosome. And that is also a homologous pair because they're the same size and the same centromere position. If we look at the cells on the right, so notice the cell on the top. It has a big pink and a smaller blue. Are those a homologous pair? Answer is no. They're not the same size. They don't have the same centromere position. So because they're not a homologous pair, they are going to be haploid, right? They don't have their homologous pair, therefore they will be haploid. So question number 5. How many chromosomes are in this cell? Is it red, 24, yellow, 12, green, 6, blue, 3, or purple, 2? So again, you will post this answer on Canvas and the answer will be provided for you later. So how many chromosomes are in this cell? Remember that the black represents the centromere. So you need to think about what are these two that are held together called, etc. Question number 6. How many sister chromatids or DNA molecules are in this cell? Red, 24, yellow, 12, green, 6, blue, 3, or purple, 2. So again, post your answer on Canvas. How many homologous pairs of chromosomes are in this cell? Same answer choices. So how many homologous pairs are in there? And then just to follow up, are those cells, is this cell diploid or is this cell haploid, and why? So that's something to think about. So biological problem, what would happen during fertilization without meiosis? So without that reductive cell division. So again, the purpose of meiosis, reduce chromosome number in half. Meiosis will start with a diploid cell, a cell that is 2N, and these will be specialized somatic cells in the testes for male and the ovaries for females. And so those specialized cells in the reproductive organs, those somatic cells are going to undergo meiosis. And the job is to reduce chromosome number in half. So the cells start with 46 chromosomes, but through meiosis, when they reduce chromosome number in half, the egg will be haploid. It will have 23 chromosomes. The sperm will be haploid. It will have 23 chromosomes. And then fertilization will restore that diploid number, meaning when the egg with 23 comes together with the sperm with 23, the zygote is going to have 46. And then that diploid zygote, that fertilized egg, is going to undergo mitosis and development. Because remember that mitosis is going to be used to produce multicellular adults. So again, all the cells in our body have the same DNA because they arose by mitosis and development. So we're going to look at an overview of meiosis. And meiosis is going to start with one diploid cell with unreplicated chromosomes. Again, diploid means di means 2. They have their homologous pair. So if we look at the top cell, this would be a cell in G1. We have our homologous pair, the one from dad and the one from mom. They are unreplicated at this point. At this point, every chromosome is one DNA molecule. Then the cells are going to go through S phase. At S phase, chromosomes are going to duplicate and you end up with your sister chromatids. And so at this point, we now have still a diploid cell because it still has the homologous pair, but with duplicated or replicated chromosomes. At this point, one chromosome is made up of 2 DNA molecules. Now, one of the unique things about meiosis that does not happen in mitosis is that in meiosis, we have homologous pairs pair up. So they will actually pair up and they will do what we call crossing over, which you'll see in a minute. but homologous pair in meiosis. That does not happen in mitosis. What happens in meiosis is that we have 2 rounds of division. 2 rounds of division, meiosis I, meiosis II. Meiosis I is going to have PMAT, so Prophase I, Metaphase I, Anaphase I, Telophase I. In meiosis I, it's going to be that the homologous chromosomes separate. So remember back to mitosis, we didn't have homologous chromosomes ever separate. We only had sister chromatids split. But in meiosis, remember that the goal is to reduce chromosome number in half. And to do that, we need the homologous pairs to split. So notice that after meiosis I, are the cells haploid or diploid? So think about that. They are haploid because they do not have their chromosome pair. They don't have their homologous pair. So these cells are already haploid. However, they are still duplicated. So we need to go through another round of division. And so in meiosis II again is going to do PMAT, prophase, metaphase, anaphase, telophase. Meiosis II is going to be just like mitosis. So now we're going to have the sister chromatids separate. So those duplicated chromosomes are going to separate. The centromere split. And what we end with is 4 haploid cells with unreplicated chromosomes. If you look at these 4 cells that have resulted, notice they're haploid. They don't have their homologous pair. And if you look at chromosome number, notice the cell at the end has 1 chromosome. We started with 2 chromosomes. So this has effectively reduced chromosome number and DNA number in half. The way I remember which one splits when is meiosis I, homologous chromosomes separate first. Meiosis II, sister chromatids separate. The way I can remember that is alphabetical order. So H comes before S. So homologous chromosomes separate first, sister chromatids separate second. So applying your knowledge, so human cells have 46 chromosomes. At the end of G1, how many chromosomes are present in one cell and how many DNA molecules are present in one cell? And so we're going to go through and fill in that table just like we did for mitosis. So what you're looking at is we have our cell in G1. And in G1, remember that we're going to have our homologous pair. The one from mom is pink. The one from dad is blue. It's still in a nucleus. At this point, we have 2 DNA molecules and 2 chromosomes, right? Each chromosome is its own DNA molecule. So in humans in G1, we have 46 DNA molecules and we have 46 chromosomes. Now, if we go to S phase, remember that S phase, we are going to duplicate our chromosomes. So we're going to make our sister chromatids. The 2 pinks are now together. That is 1 chromosome, but now 2 DNA molecules. The blue one is 1 chromosome, but 2 DNA molecules. So notice that my chromosome number is still 2, but my DNA number is 4 because I've duplicated my chromosomes. So if we fill in our table, SG2, how many DNA? 92, because we replicated our chromosomes, but it's still 46 chromosomes because we haven't changed our chromosome number. So now we're going to walk through and talk about the stages in meiosis. And we're going to start with prophase I. Prophase I, chromosomes coil and become compact. Nuclear envelope is going to break down. Spindle is going to form. So that first part is very similar to what happens in mitosis. However, meiosis has something unique that happens that does not happen in mitosis. And that is that in prophase I, homologous chromosomes come together as pairs by synapsis. They're going to actually connect with one another. Each pair with 4 chromatids is now referred to as a tetrad. Tetra means 4. They're 4 DNA molecules. Non-sister chromatids exchange genetic material by crossing over. Now, why do we do this? Well, by crossing over, we get more genetic variation, meaning that you could pass on a chromosome to your offspring that is partly from your mom and partly from your dad. You don't have to just give your offspring your chromosome from mom or your chromosome from dad. It actually can be a mixture. And that's because of crossing over. If you got crossing over between sister chromatids, would that lead to any genetic diversity? Answer is no, because sister chromatids, remember, are exact copies. So this has to happen between homologous pairs, and it has to happen between non-sister chromatids. And so you can see at these sites of crossing over, they have exchanged genetic information. And so this is something unique that is going to happen in prophase I. That does not happen in mitosis, because in mitosis, we don't want genetic variation. We simply want to make an exact copy. So if we look at our cell that's going to be in prophase I, so notice that we have our homologous pairs pairing up, and they're forming that tetrad. Those are the 4 chromosomes together. You can see that we've had a crossing over event here, because this one chromosome is pink on the top, blue on the bottom. This one is blue on the top and pink on the bottom. So those homologous pairs have now crossed over, and they have exchanged genetic information. Similarly, in prophase I, we're going to have the nuclear envelope break down. We're going to have the spindle form. So in our prophase I cell, we still have our 4 DNA and our 2 chromosomes. Nothing has happened in terms of DNA number, because all we've done is we've matched up homologous pairs and we've crossed over. Chromosome number and DNA number stays the same. So in prophase I, our number of DNA molecules is still 92, and our number of chromosomes is still 46. Nothing has changed in that regard. Then we go to metaphase I. So you might recall in mitosis, in metaphase, the chromosomes line up single file with sister chromatids facing opposite ends. Meiosis I is a little bit different in that in metaphase I, it's the homologous pairs that line up at the metaphase plate. So the tetrads will align at the cell equator. So the homologous pairs are what are going to be separating in meiosis I. And so that's why when they line up, the tetrads line up together. So if we look at our cell that is in metaphase I, again, all we've simply done is move them in the cell. The centrosomes are at opposite ends of the cell. The homologous pairs are going to line up at the metaphase plate so that one homologous pair faces one way, one faces the other. Notice my DNA number is still 4 in that cell, and chromosomes are still 2. So in a human cell, we still have 92 DNA and 46 chromosomes. Then we come to anaphase I. Anaphase I, chromosomes are going to separate, but in this case, it's going to be the homologous pairs. So homologous pairs are going to move to opposite sides of the cell. Sister chromatids remain together. The sister chromatids stay together. And this is in contrast to mitosis, where the sister chromatids migrate as a pair instead of splitting up. So meiosis is going to have both sister chromatids move to one end. So in anaphase I, this is where our homologous pairs are going to separate. So this pair goes this way, this pair goes the other way. Notice my DNA number is still 4. I still have 4 DNA molecules, and they are still 2 chromosomes. Because sister chromatids did not split, chromosome number doesn't change at this point. So if we fill in our table, right, all we're doing is moving things in the cell. We still have 92 DNA, and we still have 46 chromosomes. Then we come to telophase I. Telophase I, duplicated chromosomes have reached their pole. A nuclear envelope will form around the chromosomes in some species. Not all species do that, because you just have to break them down again when you do meiosis II. Each nucleus has the haploid number of chromosomes, because homologous pairs are what will separate. Sister chromatids stay together, but homologous pairs separated. So when the homologous pairs end up in opposite cells, now the cell is haploid. It has half of its chromosome number. So if we look at telophase and cytokinesis I, we have now separated our homologous pairs, and they are in separate cells. So notice that the daughter cells, we end up with 2 at this point, and those 2 cells are going to be haploid. They have half their chromosome number, because notice they don't have their homologous pair. Homologous pairs split. So in these cells, right, compared to the cell before, this has one chromosome. So each cell has one chromosome, but the chromosomes are still duplicated. They still have two DNA molecules. So they're haploid because they're missing their homologous pair, but they have not reduced DNA number in half. So if you look at the end of telophase 1 versus G1, G1 had two DNA and two chromosomes. At the end of telophase 1 and cytokinesis 1, we still have two DNA and one chromosome. So this is why meiosis I is not finished yet, because we have halved chromosome number, but we have not halved DNA number because sister chromatids are still together. So if we fill in our table for humans, for telophase and cytokinesis, right, our number of chromosomes is going to reduce in half to 23, which means that if they're still duplicated, we have 46 DNA. So we have 46 DNA and we have 23 chromosomes. So our chromosomes split, so chromosome number reduced in half, but they're still duplicated, which is why they still have 46 DNA. We're not yet finished because we need to reduce DNA number. And so that's why we're going to go into meiosis II. So meiosis II follows meiosis I without chromosome duplication because the sister chromatids are already there. They've already duplicated. Each of the two haploid products enter meiosis II. So let's walk through the steps. So again, just like meiosis I, we have PMAT in meiosis II. So we'll start with prophase. So chromosomes coil and become compact. If the nuclear envelope had formed in the last phase, it would break down in prophase II. So if we look at our table here, prophase II, all we're simply doing is getting ready to divide in prophase II. Nothing has changed in terms of DNA number or chromosome number. So in our table, prophase II, we are still going to have 46 DNA and we still have 23 chromosomes. We go to metaphase II. Metaphase II, duplicated sister chromatids are going to line up along the metaphase plate. Notice that this part is going to be similar to mitosis, right? Because in mitosis, they will line up single file, getting ready for the homologous or the sister chromatids to separate. So in anaphase II, sister chromatids are going to separate just like they did in mitosis. And so those sister chromatids are going to separate and they're going to go to opposite poles of the cell. So if we look at our table and we look at metaphase II. Metaphase II, we have just moved them to the middle of the cell. We have not changed DNA number. We have not changed chromosome number. So if we fill in our table, we still have 46 DNA and we have 23 chromosomes. Now let's look at anaphase II. Anaphase II, sister chromatids are going to separate. They're going to go to opposite ends of the cell. So now in the cell that's in anaphase II, remember that in metaphase we had two DNA, but they were one chromosome. When the sister chromatids separate, our DNA number stays the same. We still have two DNA molecules, right? We have 1, 2. But because they're not held together at the centromere, that is no longer one chromosome. That is two chromosomes. So our DNA number is 2 and our chromosome number is 2. So if our DNA number is 46 for anaphase, that means that our chromosome number at this point is also 46 because every DNA is now its own chromosome temporarily until the cell divides. So telophase, chromosomes have reached the poles of the cell. A nuclear envelope forms around the chromosomes. And with cytokinesis, we end up with 4 haploid cells. And those 4 haploid cells are unreplicated because sister chromatids had separated. So let's fill in our table. So if we go to our telophase II and cytokinesis II, once those divide, we end up with 4 cells. The 4 cells are haploid and they are unreplicated. So notice what happens is that this gamete, this cell, is going to be one DNA and one chromosome. It is now half of what we started. Remember that the cell at the top here has 2 DNA and 2 chromosomes. But now we have 1 DNA and 1 chromosome. We have reduced chromosome and DNA number in half. So in our table, if we were at 46 and we reduce it in half, we have 23 DNA and 23 chromosomes.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase I, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So this is comparing mitosis and meiosis. So in both cases for mitosis and meiosis, they both start with interphase. And in interphase, we start with a diploid cell. So notice the cell in the middle. This is diploid. It has its homologous pairs. And this diploid cell is unreplicated. In both cases, it undergoes S phase. S phase is going to have the DNA duplicated, and we end up with sister chromatids. Now, in the case of mitosis, homologous pairs never pair up. Yet in meiosis, they will pair up and they will form tetrads and they will cross over. So one of the things that's different between them, meiosis has crossing over, mitosis does not. That is because meiosis's job is to create genetic diversity, whereas mitosis is to make an exact copy. Now, when we go to mitosis, when we get to metaphase, we're going to have the chromosomes line up single file along the metaphase plate, and they're going to get ready to split sister chromatids. So in mitosis, we are only splitting sister chromatids, and what we end up with is we end up with two daughter cells that are exact copies of the parent. Notice that the daughter cells in mitosis are still diploid. Meiosis is used for a different purpose. Its job is to reduce chromosome number in half so that those gametes, those cells at the end, the gametes can function in sexual reproduction. So in meiosis, we have 2 rounds of division. First round of division, we are going to have homologous pairs separate. So again, alphabetical order, homologous pairs first, sister chromatids separate. Second, so metaphase 1, now we have the homologous pairs line up at the metaphase plate. They do not line up single file. Instead, they line up as pairs. So that in meiosis 1, homologous pairs are going to separate. The cells at the end of meiosis 1 are going to be haploid. They do not have their homologous pair, yet they are still replicated or duplicated because they still have their sister chromatid. This is why they have to undergo meiosis 2. Meiosis 2, we are going to separate the sister chromatids so that when we're finished, we end up with 4 daughter cells. And those 4 daughter cells are haploid. They do not have their homologous pair. They have reduced their chromosome number and DNA number in half. And now they're ready to function as gametes. So let's talk more about this. Question number 8 to post on Canvas. Which characteristic is similar for mitosis and meiosis? So what is something that is similar? Red, 1 duplication of chromosomes. Yellow, 2 divisions of chromosomes. Green, pairing of homologous chromosomes. Blue, exchange of genetic material by crossing over. Or purple, none of the above. So you'll post your answer on Canvas. Question number 9. Which stage of meiosis is similar to mitosis? Is it red, meiosis 1, yellow, meiosis 2, green, both stages of meiosis, or blue, neither stage of meiosis? So again, post your answer on Canvas and the answers will be provided once the due date closes. So I have a table to compare and contrast mitosis and meiosis. So in terms of mitosis, DNA replication, we're going to have DNA replication occur once during interphase. That is going to occur in S phase. Now, meiosis, we have 2 rounds of division, yet DNA replication only happens once and during interphase. So that is something that is similar between them. Number of divisions, meaning the number of times we go through PMAT. Mitosis, 1, PMAT 1 time, whereas meiosis is going to be 2, PMAT twice, right? We have Prophase I, Metaphase I, Anaphase I, Telophase I. Prophase II, Metaphase II, Anaphase II, Telophase II. Synapsis of homologous chromosomes, meaning crossing over. Does that happen in mitosis? Answer is no, because we're trying to produce genetic copies. So does not occur in mitosis because crossing over is going to provide genetic diversity. Mitosis is used for sexual reproduction to make exact copies. So crossing over does not occur in mitosis. However, it does occur in meiosis. And when it happens in meiosis, it happens during prophase I. So it occurs during prophase I with crossing over between non-sister chromatids of a homologous pair. So chromosome number 1, for example, from both mom and dad, can come together and they can cross over between non-sister chromatids. In mitosis, number of daughter cells and genetic composition. So how many cells does mitosis produce? Mitosis is going to produce 2, each diploid and genetically identical to the parent. In the case of meiosis, right? Meiosis, its goal is for sexual reproduction. It's going to have 2 rounds of division, which means that it ends up with 4 cells. Each cell is haploid. They have reduced chromosome number in half, and they are genetically diverse from the parent cell and from each other. So for each haploid, genetically different. Roles in the animal body. So for mitosis, It enables multicellular adults to arise from a zygote, right? So it allows multicelled organisms to form, produces cells for growth and repair, and then not for animals, but just for other organisms. And in terms of other organisms as well, mitosis is used for asexual reproduction without a parent. Meiosis plays a different role. The role is to produce gametes, those are the reproductive cells, reduce chromosome number by half, and introduces genetic variability, so makes genetic diversity. And so this is just to summarize, comparing and contrasting mitosis and meiosis. So meiosis provides 3 sources of genetic variation. And this is why offspring are not genetically identical to either parent or to the siblings. And so let's take a look at how this works. The first is that when the homologous pairs line up at the metaphase plate, the homologous pairs line up independently. We call this independent assortment. So each homologous pair lines up independently of any other homologous pair. And what I mean by that is that it's not that the chromosomes communicate and say all the ones from mom on one side, all the ones from dad on the other. Doesn't work that way. It's completely random the way the chromosomes line up along the metaphase plate. And so all combinations are therefore possible. So let's look at a simplified example. And in this case, we have 2 homologous pairs. In scenario 1, simply by chance, the ones from dad line up on one side and the ones from mom line up on the other. So during meiosis I, when homologous pairs split, the one from dad ends up in one cell and the ones from mom end up in the other. Then when we go through meiosis II and sister chromatids separate, right, these 2 are going to separate, these 2 are separating. So the gametes will have a large blue and a small blue, or a large red and a small red. So those are 2 different gametes that can form. If, on the other hand, they line up like this, so that the large blue is next to the small red, and they line up that way along the metaphase plate. When those chromosomes separate, the homologous pairs separate, notice that the cell is going to have the large blue and the small red, and the other cell is going to have the large red and the small blue. So that when sister chromatids separate, right, we end up with gametes that have the large blue and the small red, or the large red and the small blue. And so notice that there are 4 possible types of gametes that can be produced just by having 2 homologous pairs. Now, think about us. How many homologous pairs do we have? We have 23. So if we look at the number of combinations that we have, the number of possibilities is 2 to the n power, where n is the number of homologous pairs, meaning the number of haploid chromosomes. So in humans, since we have 23 pairs, it's 2 to the 23rd power. And what that comes out to is just by independent assortment alone in humans, there are approximately 8.4 million different combinations. And that's in each parent. Mom has that many probabilities. Dad has that many probabilities. And so just by independent assortment alone, 8.4 million different combinations. That's a lot of genetic diversity simply by independent assortment. The random way in which homologous pairs line up at the metaphase plate. Next, we have our crossing over. Again, crossing over is used to provide genetic diversity, and this happens when we get the shuffling of genes on the chromosomes during Prophase I. So again, remember that in Prophase I, homologous pairs are going to match up. And when they do, they're going to cross over. And this allows for new combinations of traits. So I gave an example here. So on the left, we have the maternal chromosomes. Notice I colored them in red. And on chromosome 15 are genes for hair color and eye color. And so let's say we have mom, and mom has a chromosome, And that chromosome has blonde hair, that's that yellowish orange color, and blue eyes. Now, the two red, remember, are the sister chromatids. They're exact copies of one another. So at the same position on the sister chromatid, where we have blonde hair, the other one will also have blonde hair. Where we have blue eyes, this will also have blue eyes. If we look at the combinations from dad, dad has the brunette hair, so brown hair, which is colored black, and he has eye color brown, so brown eyes and brown hair. So if we didn't have crossing over, what that means is when we give the chromosomes to our kids, either you would give your chromosome that is blonde hair, blue eyes, or brown hair, brown eyes. But it doesn't necessarily have to be that way, and that's because of crossing over. We get the shuffling of these genes. And so if we get crossing over where that black X is, we're going to end up with new combinations. So notice that what happens is that the one on the left, this red one, has the blonde hair and the blue eyes. But because of crossing over, this other sister chromatid now has blonde hair and brown eyes. That is a new combination. Prior to that, there was no chance of giving that to the offspring. Either the parent would give the blonde hair blue-eyed or the brown hair brown-eyed. But because of crossing over, now it is possible that they could give the chromosome that has blonde hair and brown eyes, or because of reciprocal changes, we could have brown hair and blue eyes. That again is a unique color, right? A unique combination, the brown hair and the blue eyes. Or you could give brown hair, brown eyes. So crossing over allows us to get new combinations that weren't possible before. And so this introduces more genetic variation. And then the last source of genetic variability is from simply random fertilization. A female will produce eggs with 8.4 million different combinations of chromosomes. Again, just by independent assortment alone, that doesn't even take into consideration crossing over. A male will produce sperm with 8.4 million different combinations. And so in fertilization, we have a random sperm fertilizing a random egg. First one in wins, right? And so because of this random fertilization, if you think about all the different possibilities, if the egg has 8.4 million different combinations and the sperm has 8.4 million different combinations, by random fertilization, there are 70 trillion unique combinations possible. So think about that for a minute. 70 trillion. This is why you and your siblings are not identical, unless you are an identical twin. And so identical twins are the exception to that, because what ends up happening in identical twins is that it starts as one zygote. It starts as one fertilized egg. But early in cell division, it actually splits into two embryos. And when it splits into two embryos, because it came from the same fertilized egg, both of those are going to be identical. So identical twins are the exception. Otherwise, if you're talking about random egg versus random sperm, you and your siblings might look similar, but you're definitely not going to be identical. There's 70 trillion possibilities, and the probability that you get all 70 trillion in common is not very likely. So if you think of fraternal twins, let's say you see a boy-girl twin. If you see a boy-girl twin, can that be identical twins? Answer is no. Can't be identical twins because one has XX sex chromosomes and the other one has XY. So you know right away that they're not identical because they're different genders. So that is just like any other set of siblings, because fraternal twins are when a woman produces two eggs and both eggs get fertilized independently. And so again, that's still going to be random fertilization. And so fraternal twins are no more alike than any other set of siblings because they're both two different eggs and two different sperm. So the genetic diversity brought about meiosis and sexual reproduction is responsible to a significant extent for the great diversity of life forms seen in the living world today. And evolution is spurred on by the differences among these offspring. And meiosis and sexual reproduction ensure such differences because they introduce more genetic diversity. By contrast, asexual reproduction, as seen in bacteria and other organisms, produces organisms that are exact genetic copies or clones of the parental organism. So if you think about it, right, sexual reproduction is going to be used to generate diversity. Why is that important? Well, because if you have diversity within a population, and let's say some disease comes into that population, if everything in the population are clones and they're all exact copies, the disease could wipe out that population very quickly. However, if you have genetic diversity, you might have some people that are resistant and some people that are not, in which case that will keep the species going. And so meiosis and sexual reproduction is used for genetic diversity, which is eventually going to lead to things like natural selection and evolution. So if genetic diversity is so useful, meaning it has advantages, why even have organisms that do asexual reproduction in the first place? Why even have organisms that don't do sex, meaning sexual reproduction? Well, couple reasons that it's beneficial to do asexual reproduction. Asexual reproduction produces very large numbers of offspring. They give a lot of offspring very quickly. Organisms don't have to invest in finding or courting a mate, and they're going to be used to preserve beneficial traits in a stable environment. So if that environment is stable and that organism is well adapted to that environment, well, obviously then in that case, it is beneficial to keep that trait in that environment because that organism is well adapted. And so in that case, asexual reproduction would be favored. So here are some examples of different types of asexual reproduction. So we have amoebas, which are our protists. We can have sea stars, which do fragmentation. So a piece of the organism kind of breaks off and gives rise to an entirely new organism. We can have plants that we do grafting. And so there are organisms that can do asexual reproduction, and then there are organisms that do sexual. And so the last part of this lecture is going to be looking at alteration of chromosome number. So nondisjuncture is the failure of chromosomes or chromatids to separate during meiosis. So we don't have proper separation. During meiosis I, if nondisjuncture happens in meiosis I, all of the offspring, all of the gametes, will end up with an abnormal number of chromosomes. So notice what happens here. So in this case, the top homologous pairs, those separate properly, but nondisjunction happens with this bottom pair. Both of those go to one side, nothing goes to this side. So notice that over here, this one is missing that chromosome. Over here, this one has the extra. So what you end up with is you get 4 gametes, none of which have the right chromosome number. This bottom one is N+1, meaning it has its haploid number plus 1 extra. This one, haploid number plus 1 extra, haploid number -1. It's missing one of the homologous pairs. Same thing with this one. So if we're looking at nondisjunction, it's going to be more severe in meiosis I, because in meiosis I, all of the gametes are going to be affected. Now, if nondisjuncture happens in meiosis II instead, so meiosis I happens, homologous pairs separate just fine. However, we get our nondisjunction in meiosis II. Notice that at the cell on the far right, since we don't have nondisjuncture, those are going to produce offspring, or gametes, I should say, gametes that have the correct chromosome number. In the cell where the sister chromatids didn't split, those gametes, though, are going to have an alteration in chromosome number. One of them is going to have 1 extra, one is going to be missing 1. So notice that the nondisjuncture is less severe when it's in meiosis II, because not all gametes are being affected. Half of the gametes are normal, and the other half are being are going to be different. Now, if we get fertilization with those gametes that have an abnormal chromosome number, that then leads to offspring with abnormal chromosome numbers. And so let's take a look at that. So if we look at a karyotype, remember that that is when we can look at the chromosomes. Aneuploidy is an abnormal number of a chromosome, meaning it's not a multiple of n. You could have an extra one chromosome, you could be missing one. So trisomy, tri means three copies of any one chromosome. So this would be 2n plus one extra. So an example of this that you've all probably heard of, trisomy 21, which is Down syndrome. That results from having an extra copy of chromosome 21. Monosomy, only one copy of one chromosome. So 2n minus 1. Monosomy is not very likely. Usually that is lethal if you are missing an entire chromosome, except especially in sex chromosomes, which you'll see. That's the exception to that. In terms of polyploidy, poly means many. So more than 2 complete sets of a chromosome. So the organism could be 3n or 4n. So they have more than just a homologous pair. They may have 3 of each chromosome or 4 of each chromosome. And that you see more commonly in plants. So an aneuploid condition is Down syndrome. Again, that's due to trisomy 21. And in trisomy 21, they have an extra copy of chromosome 21. So if we look at the karyotype, you can see that in fact, this person has one extra copy of chromosome 21. Now, although chromosome 21 is one of the smallest human chromosomes, it severely alters an individual's phenotype. Most cases of Down syndrome result from nondisjuncture during gamete production in one parent. So usually that is meaning that usually the egg or the sperm has nondisjuncture. It's not something that happens in both parents, but usually the result of nondisjuncture in one parent. Typically, more commonly, that nondisjuncture is more associated with the egg. And so the frequency of Down syndrome correlates with the age of the mother. And so this may be linked to age-dependent abnormalities in the spindle checkpoint during Meiosis I, which leads to nondisjuncture. So in females are born with all the eggs she's ever going to have. Her eggs are arrested in meiosis, meaning they've started the process, but they haven't finished it. So when females are born, they have all the eggs that they're going to have. Once they reach puberty, they start to ovulate, meaning they start to release the egg. Each month, one egg is going to be released. So the longer those eggs sit in the ovary, the longer those cells sit in the ovary, a lot of times what you see is that you get this breakdown in those checkpoints. Remember that the checkpoints are supposed to make sure that everything completed properly before going to the next step. However, if those eggs have been sitting for a long time in a state not dividing, sometimes it's more difficult to activate them properly and nondisjuncture occurs. And so the age of the mother seems to have a greater effect in the incidence of Down syndrome than the age of the father. The age of the father tends not to play as big of a role. And males start producing gametes at puberty. So it's different for females and males. Females are already born with all the eggs that she's ever going to have. Males continually produce their sperm once puberty hits and every time after that. So this is showing you the incidence of Down syndrome correlating with the age of the mother. And so this is the number of infants with Down syndrome per 1,000 births. So notice that when we look at age 20, 25, and 30, the probability of having a child with Down syndrome is fairly low. However, once we get to 35 and then notice even 40, even more so, notice that the incidence starts to go up. When you get to 45, it gets even higher. When you get to 50, it gets even higher. And so when you're getting to 50, you're almost at 10%. So when we get to age 50, the incidence of Down syndrome goes much, much higher. And so this is just showing you that there is a correlation with Down syndrome and the age of a mother. And that's why when women get to about 40, if you get pregnant after that, it's typically considered to be higher risk, and they will monitor you more closely because there are more risks associated with having a pregnancy later in life. Now, in terms of trisomy, trisomies of other chromosomes also increase in incidence with maternal age, but it's rare for these infants with these autosomal trisomies to survive for long. There are some instances of other trisomies, trisomy 18, for example, but usually the lifespan is fairly low as a result. Monosomies of any autosomal chromosome are always embryonic lethal, which means that it's never going to result in a baby being born. Nondisjuncture of sex chromosomes produce a variety of aneuploid conditions in humans, both monosomic and trisomic. So you can have monosomies in the case of the sex chromosomes. Now, sex chromosome abnormalities tend to be less severe as a result of the small size of the Y. So having an extra copy of the Y tends not to be as dramatic because there are not as many chromosomes or not as many genes on that Y chromosome. The other reason is that X chromosomes undergo what is called X inactivation. And in X inactivation, in the human female, one of the two X chromosomes becomes tightly coiled and inactive. So while she has two X chromosomes, in her cells, only one of the two X chromosomes is being expressed. So that makes that a little bit different. X inactivation is a random process, and it can inactivate the maternal or paternal chromosome. And the inactivation promotes a balance between the number of X chromosomes and autosomes. So because the X chromosome carries so many genes on it, having two copies could be too much. And so one of the two is going to inactivate. Only one of them is going to be expressed. And you'll see this later on when we talk about X chromosomes further. So this is just showing you some different abnormalities in terms of sex chromosome number. So if we look at XXY, so that is a trisomy, that is going to be male because the presence of a Y is male. And that is going to give something called Klinefelter syndrome. Typically, the symptoms of this would be males are sterile, they have underdeveloped testes, and they may have secondary female characteristics like breasts, for example. The nondisjunction can happen in the egg or the sperm, so that could be why you get an extra X. It could be from either parent. And in terms of the incidence of Klinefelter syndrome, it's about 1 in every 500 or 1 in every 1,000. So it's not as rare as some of the others. XYY. So XYY, your textbook says the syndrome is normal. However, there is something called Jacob's syndrome, and that is 1 in every 1,000 males. XO is going to be something called Turner syndrome. Turner syndrome, so that means XO means that they're missing that sex chromosome. They are monosomic. They only have one X, and that's it. And so in Turner syndrome, females who have this condition are sterile, and they typically have immature sex organs. They don't go through puberty normally like other kids their age. And so for Turner syndrome, this occurs in about 1 in every 2,000 females. You could have XXX where you inherit 3 X chromosomes. That is referred to as triplo X, right? Triplo X means that they have 3 X chromosomes. This can happen in nondisjuncture in egg or sperm. And if they are XXX, they are female. And their symptom is they might be slightly taller than average. So not typically a big deal. The incidence of this is 1 in every 1,000 to 2,000 females, so not as common. We can also, in addition to have an alteration in chromosome number, we can also have alteration of chromosome structure. And so chromosome breakage can lead to rearrangement. So if you think about, for example, if we were crossing over, it's possible that there could be mistakes made during this process. One example could be a deletion, where one whole segment of a chromosome gets deleted. Sometimes we get duplication events, where a segment of the DNA gets duplicated, and now you end up with extra copies of this. This is the case for Huntington's disease, which is a neurological disease. It's a late onset, and it has to do with these duplication events within a particular part of a chromosome. And interestingly, the number of repeats, meaning the number of times that that's duplicated, is correlated with the age of onset, meaning the more duplication events that you have, the more of these repeating segments that you have, the earlier the onset would be for this disease. Otherwise, typically, Huntington's disease doesn't come on until about your 40s or 50s. At which point you've already had kids and then pass that on to your offspring. But that can happen as a result of a duplication event, which leads to more copies of that DNA sequence. You could get what we call an inversion. And an inversion means that instead of it going in this order, this purple and then green, it could end up so that it has green and then red. That's an inversion. You could get what's referred to as a reciprocal translocation. And so what that means is that you get crossing over between non-homologous chromosomes. Remember that crossing over should occur between homologous chromosomes, but it could happen that you get crossing over between non-homologous chromosomes. And if that happens, notice what happens to your chromosomes. They end up being not the same. So this can produce genetic disorders or changes. The somatic cells could lead to cancer. And so let's look at an example of this. So here is an example of a translocation that can lead to cancer. This is a translocation event that occurs between chromosome 9 and 22. And what happens is we get this crossing over and this exchange between these non-homologous chromosomes. And this is called the BCR-ABL. And so there's a gene called BCR on chromosome 22 and this ABL gene, which is on chromosome 9. And what ends up happening is you get this nondisjuncture and you get this, crossing over, this translocation in where they're going to switch pieces of DNA. And what that does is that brings that BCR and that ABL, those genes together, and it produces a fusion protein. So if you look on the bottom here, this is a technique called FISH. And so what we're looking at is we're looking at these segments of these genes. BCR is the green, ABL is the red. So notice that these are the homologous pairs. So you have 2 red and 2 green. However, if you get this incorrect translocation, notice you end up with these fusions. And that is that the red and the green are together and the red and the green are together on these cells. Now, how might this happen? Well, you can get breaks in DNA from exposures to mutagens. So like exposure to radiation, which is high energy. That causes DNA to break more easily, which then leads to these translocations happening. And so interestingly, when BCR and ABL get put together, that is referred to as the Philadelphia chromosome. And people who have this BCR-ABL end up with leukemia, which is a blood cancer. So on the left, you have normal blood. On the right, you have leukemia. And in leukemia, you have these abnormal levels of these white blood cells. So notice there's a lot more white blood cells than they should. And so this type of cancer that goes along with the Philadelphia chromosomes is called CML, chronic myeloid leukemia. It's a specialized type of leukemia. You can also see here these translocations. So notice that this 22 and this 9, they're different because of this incorrect translocation. And so this is going to be your Philadelphia chromosome. And this can lead to cancer.